So today's talk is going to be a tag team, and after I give my brief introduction to integration of in vivo phage display and machine learning, a multidisciplinary approach to ligand receptor mapping of the mammalian vasculature, I'll introduce my colleague and friend Renata Pasqualini. By way of joint outline, there's a list of topics that we'll cover here. I'll begin by introducing phage display technology and screening very briefly, and then explain what the connection is between this project and the RCSB Protein Data Bank, outline the goals for the new collaboration that we've established that will involve Renata, Chris Markosian, who used to work for RCSB Protein Data Bank and is now an MD PhD student with Renata and her partner Wadia Rapp. This works also involves Brenda Vallat from the RCSB Protein Data Bank. And I'll talk specifically about the work that Brenda and Chris Markosian have been doing, extending PDB-XMM-CIF to support the results of the phage screening, and then describe the new database, data bank, phage screen data bank, and talk briefly about the framework that is going to support using artificial intelligence and machine learning to interrogate the results of phage screening, and that's going to involve critically Professor Sijian Wang, another member of the institute here. So let me just show what will be a familiar slide, that of a filamentous, the structure of at low resolution, admittedly, of a filamentous phage. This is a virus that only infects bacteria that can be used for displaying peptides on various coat proteins, and specifically the coat protein that's going to be used in the cases that Renata will describe the biology of in detail. It'll be the P3 coat protein, which is present at one end of the phage, with copy numbers ranging from 3 to 5 typically. The advantage of being able to encode libraries in nucleic acid and then have the libraries in turn displayed as oligopeptides on fused to these surface proteins is that you can create libraries where you can screen up to 10 to the 9 unique hepta or octopeptides. In this case, they're going to be flanked by 2 cysteine residues, which will ensure that, in fact, it's a cyclic heptapeptide, or a cyclic octopeptide. So in this way, we can display 10 to the 9 different cyclic peptide sequences on P3, and then use this as a probe to understand the vasculature of mammals, and understand the peptide binding properties of receptors that are present in the mammalian vasculature. So the way this screening exercise works is really quite simple. The phage are dissolved in water, or in normal saline probably, and then injected into the vascular tree of the animal. And by animal, I do include, when I say animal, I do include human. And there are opportunities upon harvesting tissues, more complicated in human, of course, than in animals where you can perform a necropsy. But you can recover tissues to which specific phage are bound, and you can then isolate the phage on a tissue-specific basis, and then sequence the phage DNA and understand which peptides were bound to the inner wall of the vascular tree on a organ by organ system basis, and then understand the tissue specificity of binding of these different peptides. This technology is suitable for use in man, macaque monkeys, and other model organisms. And other model organisms, all the way down to mice and rats. The connection to the PDB. So as you know, the Protein Data Bank is now in its 52nd year of operations. It was started in 1971 with just 7 protein structures, some of which are depicted here on the right. It's grown more than 30,000-fold, and as of a couple of days ago, we provided open access to more than 202,000 experimentally determined structures of proteins, nucleic acids, and their complexes with one another and with small ligands. Management of the PDB archive used to be a US-only activity, but since 2003, it's become globalized, involving data centers here in the US. So RCSP Protein Data Bank based at Rutgers, UCSD and UCSF, and also Protein Data Bank Europe, Protein Data Bank Japan, most recently Protein Data Bank China, and then two specialist data resources, Electron Microscopy Data Bank and the Biological Magnetic Resonance Bank. Those last two are for specialist resources for different technologies. It's not an exaggeration to say that the Protein Data Bank has been the vanguard of the open access data movement in biology since its inception in 1971, and it's been accredited both by the Core Trust Seal and most recently was designated by the Global Biodata Coalition as a global core biodata resource critical for the biodata infrastructure worldwide. At the RCSP Protein Data Bank, we convert global data into global knowledge through a process depicted here with this flowchart that involves the information coming in from over the life of the archive, more than 60,000 data depositors. We share responsibilities for the processing of the incoming data with our worldwide Protein Data Bank colleagues. That involves deposition, rigorous validation, and expert biocuration. Then as the Worldwide Protein Data Bank designated archive keeper here at Rutgers, we package the information up on a weekly basis to put it into the core archive, which currently has, as I said, more than 202,000 structures. These data are made immediately available to now more than 475 trusted external data resources that are free to repackage, reuse, and redistribute the data under the very permissive licensing terms on which we make the information available. Here at Rutgers, we have two websites, a research-focused rcsp.org, which serves many millions of users around the world. Thus far, we've been able to document utilization of rcsp.org at more than 200 sovereign countries recognized by the United Nations. We also have a training, education, and outreach data resource, pdb101.rcsp.org, which last year provided literally millions of page views. One of the reasons all this works is because of the very strict data representation and data model, data dictionary that we use to classify and process and ultimately archive the information coming into the Protein Data Bank on a weekly basis. It's called the Protein Data Bank Exchange Macromolecular Crystallographic Information Framework Data Standard. It's based on an earlier small molecule crystallography, chemical crystallography data standard, which was known as the Crystallographic Information Framework. The Worldwide Protein Data Bank manages the PDBX MMSIF data standard and has created a number of extensions of this, including one for small angle scattering, one for the PDB Dev prototype archive that's operated by Brynda Vallat here at Rutgers, and also the Model SIF data standard, which is used both by AlphaFold and by the Model Archive. It's this same data standard that we're going to use to take all the information that Wadi and Renata and Chris and others have accumulated over many years that's currently sitting on Excel spreadsheets and put it into a data resource that can then be made broadly available and can importantly be interrogated using tools from artificial intelligence and machine learning. So this all starts with the data standard, which Brynda and Chris have been working on for some time. And it will then be possible using that data standard with funding that we're currently seeking to assemble all of the data sets that are currently available into a new phage screen data bank. And this PSDB is going to be then linked with 3D biostructure information, both experimental and computed, and other related data from trusted sources, so that it will make it possible for artificial intelligence and machine learning methods to be used to actually interrogate the archive in the most detailed and powerful way. What will emerge from this is hypotheses as to which peptides are binding to which receptors in the vascular tree of whatever the organism is, and that those hypotheses will ultimately be testable in the lab. And the system will, of course, will be extensible so that as new and different phage screen studies are done, that information could also be brought into the phage screen data bank. Here is an example of the sort of categories of metadata information that will be used as part of the phage-CIF data standard to ensure faithful archiving of the information that's been collected already and information that will be collected from new and different screens. The PSDB will ultimately have a data warehouse, which will be wherein the phage screen data will be integrated with related information. And as I said, I've talked about 3D biostructure data, but of course we're going to be bringing in human protein sequence reference data, the Human Protein Atlas, information about subcellular localization, information about interactomes, and also drug chemical resources such as drug bank, and ultimately clinical resources such as TGCA and CBioPortal for those interested in cancer. The framework's simple. This database of bound peptides that are identified with binding to specific tissues will be used to identify lead peptides and ultimately to identify candidate receptors for these lead peptides, whether it's the heptapeptide library or the octapeptide library. Just a brief note about oxidation states. The human vasculature, like other mammalian vascular trees, is overwhelmingly oxidizing in its environment for the free phage as they're circulated. So the cysteine disulfide bridge will be formed, and these will be cyclic peptides, constraining the conformational properties somewhat. I think the opportunities here are going to be enormous, and we are anticipating success in applying for a large multi-PI grant to the NIH to establish this data resource and make it possible for Wadi and Renata to unlock the trove of data that they've already accumulated and set the stage for others to be able to contribute. So with that, I'm going to stop, and I'm going to introduce Renata. And let me just go. So those of you who can read backwards will have seen Renata's entire slide deck. I apologize for this. Okay, so for the speed readers, you can leave, but for everybody else, particularly me, I'm going to hang around. So Dr. Renata Pasqualini is a professor of medicine and chief of the division of cancer biology at CINJ Rutgers University. She's the co-founder of 2 biotechnology companies, serving as chief scientific officer for Phage Nova Bio and member of the board of directors for Embrace Therapeutics. Both companies have their homes currently in the Bristol Myers Squibb incubator, which is located in Summit, New Jersey, the old Celgene site. Dr. Pasqualini received her PhD from the Ludwig Institute for Cancer Research branch in Sao Paulo, Brazil, where she was born. And thereafter, she did postdoctoral training at Harvard Medical School and at the Burnham Institute in La Jolla, California. In addition to her activities as principal investigator and head of a large research laboratory that she runs jointly with Wadi Arab, to whom she's also married. She's previously worked at the University of Texas MD Anderson Cancer Center. She's also a board member, reviewer, and chair of multiple review panels for the National Institutes of Health and the Department of Defense, along with several other American, Asian, and European foundations that support basic and clinical research. She's a referee for many top journals featuring cutting-edge papers and technology. And to date, she's published more than 200 peer-reviewed publications and is also the author of multiple issued patents, both here in the United States and in other geographies. So, Renata, it's great to be partnering with you on this project. Thank you very much for coming.
Hi, everyone. Thank you for coming. Thank you, Stephen, for the invitation, for having me. And I'm thrilled to be here in such a distinguished company to tell you about our work. So I think Stephen already disclosed a few of the conflicts of interest that I wanted to cover, namely the technology that's being licensed to biotech companies in which we're heavily involved towards developing some of our discoveries into real therapies in the clinic. And also the fact that I run the lab with Wadi Arap. We've been married for 30 years, run the lab together for 25. And given that we're still alive and still married, you can tell he works fairly well. He's an incredible doctor, an oncologist who sees patients on a weekly basis and a brilliant, brilliant scientist. So I'm very fortunate. I also want to acknowledge our team, both the alumni and the current configuration of the laboratory. I often speak so quickly, I don't get to say their names specifically, but we've been blessed with an enormously talented and devoted group of folks that have really made all this possible. As you see, the technology is very broad, the opportunities are many, and without such great people, we wouldn't really be able to bring it to fruition in the way we have and intend to continue to do. And I decided to follow Stephen's kind introduction to not only the system we utilize, the phage in and on itself as a very flexible entity for bioengineering in terms of manipulation of the genome and the capsid proteins. So I decided to divide my part in three to give you a broad idea as to the foundation of our program, the overreaching theme, which is the ligand-directed targeted delivery of therapies or imaging agents or whatever payloads of interest, the vascular zip code system that we were able to uncover through in vivo phage display in and on itself back in the mid-90s. And then examples of two programs that we brought all the way from basic discovery, screening a combinatorial library all the way to a phase 1 trial independently, thanks to philanthropy funding that we received at MD Anderson, and an update on the human vascular map, as we call it, which is beyond omics, as I've been saying, and the opportunity we plan to explore with Stephen and a team of very accomplished investigators to bring the project to the next level as we expand our capabilities to mine the data and understand the vascular system and the specificity of these receptors in a more sophisticated way. So following up on what I just said, it became clear to us, although at the time it wasn't well accepted or even understood at all, that there's a fairly broad level of diversity within the vasculature in the human body that includes normal tissues and diseased tissues. So you can think about it as an address that drives the trafficking of proteins and cells and tells them where they need to go, both physiologically and pathologically in terms of a functional directing system. And those markers are accessible to the circulation. And this is the way we find them, by introducing these combinatorial libraries intravenously and recover the phage that ultimately enrich and localize to the blood vessels in the kidney, the lung, the brain. And then there's a further level of complexity that emerges when you have abnormal vasculature or pathology undergoing enacting changes in blood vessels, namely oncology, tissue remodeling, trauma, inflammation, and the like. So all these We have extensive literature outlining this discovery process, and also mechanistic data that emerges from studying the ligand receptor pairs. Just a caveat here showing you this beautiful imaging in which we can probe diversity even at the intra-organ level by using laser capture microdissection. So this is an example of a screening that we did in which we recover phage from pancreatic islets and demonstrated that the specificity was really striking in the sense of seeing the phage as red dots here by a high magnification imaging and not at all binding to the rest of the pancreas. So I would say the diversity is limitless as to what you can discover depending on how you plan the screening. A 3D image of the phage here. I'm sorry, I should use this for the folks that are watching in Zoom. I apologize. So the 3D representation of a bacterial phage, as Stephen mentioned, this is a virus that evolved through time to infect only bacteria. And there are two types. They can be lytic, in which case they can be used as antibiotics, or lysogenic, which is the type of phage that we use to construct our libraries. So if you engineer the genome, you can display peptides on P3 or on P8, and you have a lot of diversity in terms of what you can probe as far as protein interacting sites. The original application, we didn't invent this library system. As a matter of fact, this was brought about in the mid-80s, and it was highly utilized by many folks to map protein interacting sites. Original applications include, but not limited to, mapping antibody binding sites. So if you expose the library to an immobilized monoclonal, the peptides you recover will look like the epitope towards which the antibody is directed against. So it is fairly well validated. What we brought about was the ability to screen these libraries in vivo, and of course, discover a whole new level of specificity for targeted delivery, and again, understanding the complexity of receptor systems in specific microenvironments that are well defined based on the tissue specificity of the ligands. So there are three arms that we've been pursuing over the years. Of course, using the combinatorial libraries in vivo, in mice, rats, swines, all the way to humans. At MD Anderson, we already approved an IRB protocol to be able to do this directly in cancer patients, in which we infuse the library after consent is given, and then we can mine biopsy and autopsy material from multiple organs and cancer lesions. So that expanded our database substantially. And upon prioritization of two ligand receptor pairs, one that's prominent in bone metastasis and another one in the vasculature nourishing white fat, we carried out two clinical studies, and I'll show you the results in a minute. What we brought about when we spent a brief period of time in the University of New Mexico interacting with Los Alamos National Laboratories and Sandia Laboratories was the ability to do these screenings with antibody libraries. So now you're displaying a human antibody on the surface of the phage. And this is a very powerful system because you get more drug-ready ligands, namely human antibodies that can be conjugated to cytotoxics and create ADCs. And I'll show you just a snippet of our latest one that's actually about to be filed as a pre-IND on Friday with the FDA. But back to the basics of the selectivity. Why does the screening work at all? How does it work? And why is it different, in a way synergistic to other high throughput technologies such as proteomics, genomics, and the like? As you know, when you look at the repertoires of genes or proteins and look for patterns of expression in different tissues, normal or diseased, you array everything. So you lose the microanatomical context on that process, and you're only stuck with whatever is high or down. What we do is different because the screenings happen in vivo within the structural microenvironment of the tissues. So because you're introducing the library IV, systemic accessibility of the ligand is a given. So obviously the phage is circulating. It's like mounting a camera, and the phage are looking at the blood vessels and being able to see the receptors and then interact with them, from which we can then recover the ligands and using biochemistry identify the receptor. So this happens in vivo in microanatomical context. And an interesting feature is also the fact that nearly all the receptors we map have internalizing capabilities, which we believe is what mediates the enrichment. So you can imagine the phage circulating, it hits a receptor, internalizes, it hits it again, internalizes, and that's why you get the ligand side covered by enriching the frequency of peptides you isolate based on its recognition of a specific receptor. We also map very interesting environment-dependent receptor features, and by this I mean protein complexes that come together to form a binding site, activation states, conformational states that you wouldn't necessarily be able to reproduce in vitro. And again, the accessibility is crucial. In a normal vasculature, as you see here in breast, the endothelial cells are organized, they're not fenestrate, they're very, very well behaved in terms of their composition, whereas in tumors is a completely different picture. It's not difficult to imagine the biochemical diversity that explains what you see here in terms of receptors that are present or absent depending on the microenvironment, and also the accessibility from the circulation to pericytes, tumor cells, tumor infiltrating cells, stromal cells, stem cells, and what have you. So it is very important to keep that in mind as you interpret the results. This is a collection of receptors that we have been studying. The ones in red are the ones that are more advanced in terms of developing ligand-directed therapies or imaging strategies, leveraging their specific expression and accessibility. And it's not unusual for us to identify proteins that are highly unexpected in the sense that they are known or orthodoxically described as being, for instance, intracellular, expressed in ER or Golgi, but within condition of stress, hypoxia, resistance to chemotherapy, radiation, they traffic to the surface very rapidly. So you have to really keep an open mind when you're looking at these receptors, because most people would say, this is an artifact, it's a Golgi protein, what's it doing in the surface? And it is true, because it's part of the biology that's driving that receptor to a different compartment. So that, again, if you were to just array everything and look for this, you wouldn't find it necessarily. And you see here a lot of stress response proteins appear in our screenings, particularly a GRP78 is a fascinating target that we've been studying for a while to target tumors. So very quickly, a couple of words on the two clinical trials that we were able to bring about. They are PDCs, peptide conjugates, so to speak, although they're made using Merrifield synthesis. And what we were able to introduce here was a very interesting tool that we discovered back in the Burnham, which is this peptide clock, 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 clock. This structure has an interesting property, which is upon internalization, it hits the mitochondrial membrane and it induces caspase-dependent apoptosis. Now, outside the cell, it is inert because obviously it has no access to the mitochondria. So it was, in a way, a perfect tool to combine with a peptide loop that binds to a receptor that has internalizing capabilities and is selectively present on one tissue and nowhere else. So that brought about the ability to perform targeted tissue ablation. And our rationale was, what are the two tissues that come to mind when you think about something you want to do away with? Number one, cancer. Number two, fat. So those are the two that we elected to validate first. So in the first clinical trial, we had isolated a peptide that maps to a new binding site in interleukin-11 and recognizes the interleukin-11 receptor, which is highly upregulated in prostate cancer, breast cancer, osteosarcoma, multiple types of tumors. But the interesting feature of this receptor is not only in the primary tumor, but it travels with the tumor as the tumor metastasizes. So we call it not a zip code, but more like a cell phone in a way, and is at its highest in bone metastasis. And we were at the time at MD Anderson in the genitourinary oncology department, so we had access to a lot of patients with prostate cancer metastatic to bone, so that was the target of our trial. So patients, so by hooking the IL-11R targeting peptide to the clac-clac, which is the apoptosis-inducing motif, we were able to create bone metastasis targeting peptide 11, not a very creative name, but at the time it worked. And we worked with a fantastic team of consultants, toxicologists, and clinicians to conceive of a clinical protocol to demonstrate proof of concept, particularly in biological endpoints that we could obtain through biopsy material from bone mets. And this is what you see here. So the patients enrolled in the trial had a very heavy metastatic burden of metastatic cancer to the bone, castrate resistant, they had failed all types of previous therapies they had been given, and then they were enrolled based on inclusion criteria. And so they were infused with BMTP11 for 4 weeks within a month. And then we were able to obtain biopsies and demonstrate that, yes, the drug got internalized using an antibody against KLAK, which is the killing moiety, half of the killing moiety. And that a subsequent tissue section adjacent to that showed by tunnel that the cells that picked up the drug were indeed dead. So there was demonstration of both drug localization and activity in a biopsy setting. So it was a small trial. We didn't enroll a lot of patients, but there was successful evidence of, again, the biological endpoints and a mild hint of efficacy. And then we switched to another population, which were prostate cancer patients with a heavy metastatic burden of metastasis to the bone with a very high BMI. Why? Because there's extensive literature that shows that a high BMI contributes to cancer progression and spread and metastasis in multiple types of cancer, prostate cancer being one of the major ones. Not to mention the obesity epidemic, which is alarming. You just have to look around in order to see everywhere. And it is projected that by 2035, 4 billion people, which is half of the population of the world, is going to be obese. And that's because of the prevalence of obesity, particularly in Africa and Asia. The US has always been the #1. And again, they have to create new colors here to represent the BMI levels, which are very alarming. And with obesity come all the comorbidities, atherosclerosis, diabetes, and what have you. So back to the point, we had isolated a peptide that had the ability to selectively localize to the vasculature that nourishes white fat and induce apoptosis. And by that process, you release lipids very slowly. You're not hitting adipocytes, you're hitting the vasculature. So we did a study in mice, and we were successful in observing weight loss. And then we moved to rhesus. We had access to a colony of spontaneously obese rhesus in Texas. And those monkeys were subjected to therapy with this peptide. Hooked to the clac-clac moiety, and also showed substantial weight loss, diabetic reversal, metabolic syndrome with no steatosis or dyslipidemia. And imaging studies did show a substantial decrease in both visceral and subcutaneous fat. The only adverse event was accumulation of the clac-clac moiety in the proximal tubule, which created a reversible elevation of creatinine, the code word being reversible. Which is favorable in terms of an FDA overview of the safety window. And the interesting fact that I want to close this particular part with is that when we moved to humans, we saw a migration in the effective dose of 10-fold compared to the rhesus, which brought about a much wider safety window. So now this drug can be tested, hopefully, in a pre-bariatric surgery setting or settings that are not necessarily cancer. and we hope to be able to develop this further. I want to close by giving two quick examples of how we can also use the phage as a gene delivery tool and manipulate its capsid for dual display. Stephen mentioned, and I did as well, the display of peptides on P3. You can also engineer the genome to manipulate and express peptides on P8. And in addition to that, a further level of engineering can be achieved at the genomic level, in which you introduce a promoter, a mammalian promoter, and a gene of interest, so that once the phage DNA is internalized, it can mediate gene expression in a targeted way through the ligand receptor systems I've been describing. And I give you one example here. This particular peptide was identified by its ability to target gliomas. And you see here how sharp the line is between normal brain and the brain tumor. And here's an empty phage that just passes by. It doesn't bind at all, and it doesn't trap, it doesn't localize whatsoever. So by delivering thymidine kinase, which is a suicide gene, if you introduce ganciclovir, or an imaging gene, if you introduce a tracer such as FIAL, you can do targeted imaging of brain tumors, and you can do therapy by, again, suicide gene therapy approaches. So this was very interesting proof of concept. You can also introduce transcriptionally driven imaging if you use a promoter of a protein that you know gets upregulated, again, particularly upon tumor resistance to therapies or radiation. And it's what the famous diagnosis paradigm in which you see and treat and see, and you can make more educated decisions as to what therapies the patient should receive or not receive in terms of the response that's being observed in real time. And when COVID hit, we all had to theoretically go home and shut down unless we were working on something that was related to SARS-CoV-2. So we switched the focus of the lab and started collaborating again with a very bright group of investigators, Stephen's included. And we started to work on a phage-based vaccine that could eventually be used to immunize against SARS-CoV-2. And that was well integrated with another platform that was going on in the lab already, which is the ability to screen the libraries by aerosol administration and looking for phage that you can find in the circulation through the airway. So that was a very interesting way to immunize if you were to display specific spike protein peptides on P8 or express the SARS-CoV-2 spike protein through a gene delivery system. And obviously focusing on epitopes that were less likely to be mutating upon the emergence of variants, which would be the most effective ones for vaccination. And again, this was published and was very nice line of work that we continue to pursue, led by Chris Marcosian and Daniele Stacquicini and a bunch of other folks in the lab. And this is the update, going back to what Stephen mentioned, that I wanted to finish with in terms of the database of tissues that we have is very extensive, not only multiple species, but predominantly human and rhesus monkeys. Again, from mining biopsy and autopsy material, you can get repertoires of ligands that localize specifically, do subtractions by bioinformatics, and really come up with a very informative ligand-based map of the entire repertoire of organs, again, normal and diseased. And I won't go into detail in terms of the minutiae, how we mine this data. And again, we plan to bring it to the next level as we get into a structural view of what these ligands are like, as opposed to just looking at linear sequences, which is very limiting and has been very limiting for us. And again, just to close with the latest in terms of antibody library screenings, this has been a very successful endeavor. And we have a very interesting model of colon cancer that we have access to in monkeys. So we have a number of antibodies that we recovered in these monkeys and now are translating into a phase 1 trial. These ADCs In all my years in oncology, I've never seen anything so active. So if you hook specific antibodies against given targets that we discover by selection of phages, play accessible recombinant target antibodies, SPARTA, these ADCs are just incredible. You can see this one against GRP78. One dose, you do away with tumors. Those are all PDX. We tested over 60, and the activity is just remarkable. So we really hope to be able to help patients and develop this new platform very soon. So with that, this is my acknowledgement slide. We have a relatively small group, but we continue working with our alumni and a fantastic team of collaborators to which we're very grateful and look forward to doing even more. Thank you again for having me. I'm happy to take any questions if you have any.
Thank you, Renata. So I'll just show one more slide, and then Renata and I will do the Q&A. So if you'll just bear with me. Let me see. Let me just share screen again. And here it is. Okay, I just wanted to acknowledge members of the RCSB PDB team broadly, and to find Brenda Vallat here and thank her again for the work that she's been doing on phage SIF with Chris, and to thank the NSF, the NIH, and the US Department of Energy for funding the core operations of the RCSB Protein Data Bank. And thank you, Renata. So we'll take the Q&A and look forward to the interplay. Monica.
So in the triple negative breast cancer SPARTA, do you know what receptors those are targeting?
Yes, we do. As a matter of fact, I just didn't have time to go into detail. But that happens to be a very interesting new target that hasn't been previously recognized to be expressed in a plethora of tumor types that we looked at. Happens to be a molecule called FA5. It was known to be involved in neuronal migration during development, but we found it very highly upregulated in all tumor types we looked at, including advanced tumors.
And how do you identify the receptor?
Yeah, so the receptor is FA5.
Yeah, but how do you identify the
So,
receptor?
Yes, so the receptors are identified by biochemical approaches. So if you have a peptide or an antibody, what you do is you just immobilize it and you just run an extract through, purify it, mass spec, and see what's binding to it. And then you validate it extensively through tissue microarrays, looking at human tumors, expression profiles, and the like. So that's how you prioritize the ones that are expressed in the majority of tumor types, in the majority of patients that would eventually be treated, which is likely the way to start, because you don't want a target that's very specific to a patient population or one type of cancer. So this is the approach we've been taking. And so this first one that's going to the FDA Friday is Ephrin A5 with an antibody that we pulled out from the in vivo phage display with antibodies.
Okay, I do not see any questions in the chat. Are there any other questions that people would like to ask? Greg, I can normally count on you for a question. Good, thank you. This is a man who came from Bronx Science High School, you can tell.
Yeah, that's good. Go to high school in the Bronx, come with a lot of questions. Apoptosis target, the apoptosis targeting. Sort of too fast a question. It looked like you were using a peptide that be the clac, which was, those are D-amino acids that was being used. So it was just, in terms of biochemical question, is that, I suppose you tried it with standard L-amino acids and it does not trigger apoptosis. It needs to be the D.
Correct. Correct. So just quickly, the way the clac story evolved is we were collaborating with a brilliant chemist at the Burnham at the time. And clac had been described in the literature in L form as an antibiotic. So he conceived of the fact that the membranes of mitochondria are similar to the membranes of bacteria. So maybe this peptide might induce apoptosis if it hits mitochondrial membranes. And yes, he demonstrated that. It wasn't very effective in L form because it would degrade very quickly. So he synthesized in D form, which made it very stable. And then he came to us, Wadi and I, and said, I have this really nice tool to induce apoptosis based on mitochondrial swelling, but I give it to cells and it doesn't do anything. And we said, of course it doesn't do anything because it doesn't get inside the cell. But we have the perfect tool to combine both in order to internalize through ligand receptor directed internalization, which is what we do. And that's when we came up with the ability to do this modular tissue destruction. If you send it to cancer, if you send it to fat, if you send it to lung, you would destroy that particular vascular bed based on specific internalization. And the D form is effective, but the L form is not, just because it degrades very quickly.
Monica.
Sorry. So for every one of your phage display, you have to be able to isolate your organs and then pull out the phage that's internally in it. So that's for humans, you have to be able to have access to biopsies then?
Biopsy and autopsy material. So the phage gets degraded fairly quickly, but we've been able to, in certain settings, using autopsy material to mine up to 72 hours post-infusion because the DNA lingers. So by using PCR, you can just amplify what the peptide was, even if the phage is gone and degraded. So.
Yeah, and I think the first case in which you were able to do this in a human was an individual who was passing away from myocardial infarction.
That's correct.
And
yes.
So that individual, I guess either the individual or the family,
yes,
consented to, under
autopsy,
informed consent, to the infusion of the phage and then the autopsy and the harvesting of different tissues and the isolation of the phage. It's really quite an amazing leap of faith on the part of the family to participate in this.
Yeah, and of the institution too, because at the time they were very forward thinking in terms of doing this work in patients.
Yeah.
And then Wadi worked with ethicists in order to really map out guidelines that separate this type of research from transplants. Of course, cancer patients are not eligible to be organ donors. And then just a couple of caveats. You see, now we have all these capabilities to do next gen sequencing and get millions and millions of phage. But in my day, when I first did the in vivo phage display screening for the first time, I would be lucky to sequence 12 phage in a day, by pouring the gel and loading it. None of you probably remember what that is. Yeah, it could be done even in low throughput, but today we just have such capabilities. And again, the ability to integrate this with you guys is just going to be unbelievable.
Okay, Monica.
I've done receptor screening, so I have lots of questions. So you don't actually have to cyclize your peptides because of the oxidative environment, or do you pre-cyclize your peptides?
We do. So in the phage, the way we display, as Stephen described,
right, but afterwards, when you use it as a drug,
we do cyclize, and we do see exceedingly exquisite specificity depending on sometimes we find double cyclics, and the way the cysteines pair is crucial for the specificity. So if you pair them like this, they don't work. If you pair like this, they work. So there's a lot of sophistication in terms of the conformation of the loops. And it's important that the fact that they're cyclic. For instance, if you screen linear libraries that we have in the past, you always come up with cyclic peptides anyway from the combinatorial selection. So that tells you that the loop is really the business side of the biochemical interaction.
So one's only limited essentially by the size of the library, and which is governed by the size of the peptide.
Yes.
Yeah.
And I was telling Sijan before, a lot of laboratories can't do this well. And most of the reason is they start with libraries that are not well done. So they don't have the sufficient diversity, or they just already have a bias at the time you start screening them. So the screenings don't reveal anything specific.
But the 10 to the 9 is comparable to 20 to the power 9, isn't it? 20
Correct.
20 to
So
the power
each-
7 or
Each
20 to the
amino acid on
power
each position, if you
uh,
do a good job in your library,
20 to the
which-
power 8. Yeah.
which, correct me if I'm wrong, it's supposed to cover most protein interacting sites in
Yeah,
nature.
yeah.
From a mathematical perspective, right?
Sure. I mean, there are going to be some limitations, but
Yeah.
you do what you can. Yeah.
Yeah.
Okay, let's thank Renata once again. for a
Thank you
great
so
talk.
much.
And as a reminder to the Institute graduate students, we'll be meeting in room 2.